Meta-analysis types that can be performed to analyze data or result extracted from multiple independent research or clinical or medical or healthcare studies to generate a overall or pooled effect size. There are several types of meta-analysis that can be used or performed depending on the experimental or intervention design and on the outcome of the research or the trials or the study. So single group meta-analysis is the first type. The second one is two group meta-analysis. This type of meta-analysis can be performed when there are two groups. The research designed that there will be two groups. One group is the treatment group or the intervention group or the exposure group. A second group is the control or unexposed or a reference group to compare. So then two group meta-analysis can be performed. And the third one, which is the multiple group meta-analysis, is performed when there are more than two groups. For single group meta-analysis, a proportional meta-analysis can be performed, or a means meta-analysis can be done, or correlation coefficient meta-analysis, or incidence rates meta-analysis. All this depends on the research outcome and the estimated effect size. In the first analysis, it is a proportion or a percentage. In the second one, the estimate or the effect size is the means, and in the third one is the correlation coefficient, and in the fourth one is the rate. To perform proportional meta-analysis, the outcome from the studies can be any type of a binary data that is recorded in a format of a percentage or a rate or a proportion or a frequencies. So any format of the data extracted from the studies that is reported in a percentage or proportion with a single group, then proportional meta-analysis can be performed. The proportional meta-analysis can be performed to calculate the disease or the condition prevalence in a population. It can be also used to calculate the overall treatment response rates, adverse events rates, incidence rates. So it can be either looking for the prevalence or the incidence of a disease or a condition in a population, or treatment response rates for in specific intervention, or for side effect from having a treatment with drugs or medicine. So meta-analysis can be performed, proportional meta-analysis, on data extracted from several multiple studies that report the outcome in either percentage or rates, and looking for prevalence or treatment response or adverse effect or adverse event or adverse effect of a drug. The second type of meta-analysis is the pairwise group meta-analysis. Pairwise group meta-analysis is performed when there are two groups, exposure or unexposed or control. The outcome can be continuous. In this case, the standardized mean difference can be considered and used as an effect size in which the means and the standard deviation for the exposure and the control is calculated and provided. Then the effect size, which is SMD, can be used, and there are several type of standardized mean difference, using the Hedges or the Cohen's d or the Glacius delta approach. If the outcome is a binary, and a binary outcome is an outcome in which the data have only two levels, either yes or no disease or no disease, then the effect size that is measured and can be used for two group meta-analysis is the odds ratio, the risk ratio, the hazard ratio. And finally, the third type of meta-analysis is multiple group meta-analysis. When there are more than two groups, then this analysis will be used when there are more than two groups, and it is called the network meta-analysis. So there are several different types of meta-analysis to perform, and selection of each type depend on the experimental or intervention design and the outcome, whether there is a single group, pairwise group, or multiple groups.